Hello everyone, welcome to my YouTube channel shikshak. This video will be very helpful for all who are preparing for CSIR UGC NET, Life Science, GATE, ARS NET examination. Today we will discuss about chromatin immuno precipitation assay known as ChIP assay. Also we will deal with the practical as well as the theoretical aspect of it. Chromatin immuno precipitation assay is used to detect the DNA and the protein interactions. Also to know epigenetic changes in the DNA. Suppose we have done an experiment in the lab and now we have discovered a new transcription factor. Transcription factor is a kind of protein. Now the question arises, which gene this transcription factor regulates? To what protein it binds to in particular process? And to know the exact sequence of DNA where it binds. Now let us first have an basic idea of chromatin structure. In the cell nucleus, the DNA is compacted to form chromatin. Then this chromatin is more condensed to form a chromosome. But we will mainly focus on chromatin and how the DNA wraps around the histone proteins. At these histone proteins, various type of processes takes place, namely acetylation or methylation, which either activates or deactivates certain processes. There must be some transcription factor which will help in the synthesis of enzymes which are responsible for either acetylation or deacetylation or for other type of modifications. Let us take a look on experimental procedure of ChIP assay. In the figure you can see cell is firstly lies to get the chromatin. Chromatin is a substance in the chromosome that consists of DNA and proteins. These proteins are known as histone proteins. These proteins binds to DNA to form a compact structure chromosome. Firstly, this chromatin material is lysed with the help of restriction endonucleases to get the different type of the fragments. That includes either histone bounded or non-histone bounded or only linear DNA without histones. After that, immunoprecipitation is done with specific antibodies that are specific for histones or other transcription factors. After that, centrifugation is done. After that, we will get supernatant and the pellet. In the pellet, we will get the antibody attached to the specific proteins or the transcription factors that are of our interest. And supernatant we will discard. Supernatant consists of unbounded protein or without antibody attached proteins and linear fragment of DNA. The pellet is also known as pull down fraction. This picture is just an addition to the previous experiment that we are discussing. Before digesting the chromatin with the help of specific nucleases known as micrococcal nucleases, which are obtained from the micrococcal bacteria, cells are fixed with the formaldehyde to cross-link the histone and the non-histone proteins to the DNA, then digested to get the 150 to 900 base pairs of DNA and protein fragments. Then specific antibodies is attached. The process is known as immunoprecipitation. To these antibodies, sometimes magnetic beads are attached. This helps during the centrifugation process or during the magnetic separation processes. Then high salt concentration is provided so that this protein and the DNA sequences separates out. This is the sequence of our interest, where exactly the transcription factors binds. Now the sequence is known to us where the transcription factor binds. I think this is clear to all of you right now that why we perform this assay. This picture is again an representation of how chromatin is digested with micrococcal nucleus and how the anti-tag antibodies are attached to specific proteins and different type of fragments we get. We get either linear DNA without any protein or transcription factor bound DNA with specific anti-tag antibodies in a append-off tube. And remember, this is known as pull-down fraction. To this pull-down fraction, high salt concentration is provided so that protein that were binded to this DNA get separated. Now we can use this DNA sequence or DNA fragment for performing PCR, qPCR or the microarray techniques to confirm the DNA and protein interactions. I have added these pictures just to revise the concept again. Here I have written two transcription factor TFA and TFAB. Just keep in mind TFA is specific for A sequence on the DNA and TFB is specific for B sequence on the linear DNA. In the figure you can see there is a linear DNA which have two sequences. First sequence is A sequence and other sequence is B sequence. Now we will add this linear DNA into two append-off tubes marked number 1 and number 2. To the append-off 1 we will add TFA only, and to the append-off 2 we will add TFB only. And remember, these TFA, TFB are the transcription factors. Now these transcription factors will bind to the specific sequences. As I have told you that TFA is specific for A sequence. That's why it will attach to the sequence A in the first append-off tube. And at this step we add anti-tag antibody which bind specifically to the TFA. After this we will add micrococcal nuclease that will fragment the chromatin into the three structures: linear DNA and the bound proteins. Then we centrifuge and discard supernatant and will use the pellet, which is also known as pull down fraction. This consists of transcription factor A bonded to A sequence on the DNA, and this sequence is of our interest. In the append-off tube we will add TFB, which is specific for the B sequence on the DNA. Here one thing you can observe, TFB binds to B sequence, not to A sequence, even though both sequences are present. Now we will add specific anti-tag antibody in the append-off tube. After that we will digest this mixture with the help of micrococcal nuclease. After digestion we will get three different type of fragments: linear DNA bond TFB to B sequence. After that we will centrifuge it. Then we discard supernatant. Then we use pellet or the pull down fraction. Pull down fraction consists of B sequence, which is of our interest. Let us now solve the question which appeared in the CSIR UGC NET 2014 December based on the information we have discussed. The question is, a chromatin immunoprecipitation assay was performed to determine specific transcription factor binding sites on the promoter of a gene. A pull down was done using either IgG or antibodies against CMYC. A DNA containing CMYC binding region was used as a control for PCR amplification input. Which one of the following PCR representation of DNA is correct? In the question, it is given that there is a DNA sequence that is having CMYC binding region, indicating that CMYC protein or the transcription factor CMYC will bind to the promoter of this sequence. And the second information is that pull down was done by two antibodies. First one is IgG and the second one is antibodies against CMYC. So let us try to solve this question now based on this information. Here in this question they have taken three append-off tubes. To all three append-offs they have added linear DNA. Then they have added CMYC protein or the transcription factor to all three append-off tubes. As in the question it is given that linear DNA consists of a CMYC binding region. Hence this CMYC protein or the TF factor will bind to the linear DNA on the CMYC binding region in all three append-off tubes. Next step is the immunoprecipitation, in which we add anti-tag antibodies. In the question, it is given that they have used IgG or the anti-CYC antibody in the experiment. Suppose if they have added two antibodies in the control, what only anti-CYC antibody will attach to the transcription factor, because this is specific for the CMYC protein. In the second append they have added Ig antibody, as these are not specific for the transcription factor or the CMYC protein. So no antibody will bind to the transcription factor. To the third append they have added anti-CMYC antibody, which will bind to the transcription factor. After nuclease is added and centrifugation is done. Now we have got pull down fraction for all three append tubes. In the append 1 and 3 we have got transcription factor bound antibody to the sequence. But in case of second append we haven't got any bound sequence, as no antibody was bound to transcription factor and no pull down fraction was obtained in second append. And whenever we will run a gel, we will get a band in the input or the control, and also get a band in the third append which is having anti-CYC antibody. But in the lane 2, where we have added IgG antibody, we will not get any band. Now you can observe these options. In option A, C and D, we are getting band in the lane 2, which we have added Ig antibody. Hence these options cannot be the right options. Hence the right option is answer B. In the option B, we are getting thick band in the first lane and the third lane. For your general information, thin band represents no bound protein and thick band represents a bound protein or the excess protein. And don't forget to subscribe to my YouTube channel, that is Shikshak, and do hit the like button, share and subscribe to my channel.